Body structure and production of antibody fragments. Antibodies are large Y-shaped proteins produced by plasma cells. Immunoglobulin G is the most common type of antibody found in blood. The innate immune system utilizes these proteins to identify and neutralize foreign objects, such as bacteria and viruses. Antibodies exist as a copy of a Y-shaped unit consisting of 4 polypeptide chains. Each antibody is composed of 2 identical heavy chain and 2 identical light chains distinguishable by their variation in molecular weight. Also crucial to the antibody structure is the constant and variable regions. Each antibody contains 2 light chains that are always identical and help to classify the antibody as either a kappa or a lambda type. A light chain has 2 successive domains, 1 constant domain and 1 variable domain. The constant region is identical in antibodies of the same isotope but differs in antibodies of different isotopes. The tips of each Y arm are known as the variable regions and are antigen binding sites. Each antibody has to have a specific shape in its variable region in order to bind and identify its antigen. Each antibody also contains 2 carbohydrates within the CH2 domain that acts to stabilize the structure within the immunoglobulin G molecule. The N-terminus refers to the start of a protein or polypeptide terminated by an amino acid with a free amine group. The heavy and light polypeptide chains of the antibody are held together by disulfide bonds. The C-terminus is the end of an amino acid chain terminated by a free carboxyl group. The 2 identical heavy chains each carry covalently attached oligosaccharide groups. The light chain is separated into 2 domains, the VL and the CL. The heavy chain of immunoglobulin G has several domains, 1 VH and 4 CH regions. The heavy V unit shows similarity to the V domain of the light chain, while the C units show homogeneity to each other and to the C unit of the light chain. Papain is a thiol endopeptidase. When immunoglobulin G molecules are incubated with papain in the presence of a reducing agent, 1 or more peptide bonds in the hinge region are split, producing 3 fragments of similar size, 2 Fab prime and 1 Fc. Pepsin is a non-specific endopeptidase. It is active only at an acidic pH. It is irreversibly denatured either at a neutral or alkaline pH. Digestion by the enzyme pepsin normally produces 1 Fab prime 2 fragment